Hi everyone. Today I'm going to present the lecture component of Lab 2, Introduction to Organisms. Once I share my screen, I'm going to close my little video so that the irritating rattling sound will stop for you all. And today's lab is a review and hopefully things you mostly already know, but just making sure that everybody understands the vocabulary, everybody has all the different parts of each of these different concepts and ideas. And it'll form the foundation for most of the work that we'll be doing the rest of the semester. Normally, we'll be starting to learn how to use the dissecting and compound microscopes, but since you are not having a hands-on lab this semester, you will have to hopefully already have those skills, or you will learn them in a class in the future. So you're not getting cheated out of your entire chance to learn how to use the microscope. Okay, so we're going to look at the organization of bodies of organisms. We're going to look at single-celled versus colonial versus multi-celled life. And we're going to look at the names of body parts and look at how we do sections or slices through those body parts in both animals, plants, and fungi today. So we're focusing on multi-celled life, and the multi-celled life has two characteristics that are important to define it. One is that the cells are interdependent. That means they need one another in order to function. So they aren't just stuck together. They are actually cooperating with one another. And the other characteristic of multi-celled life that's important is that the cells are of different kinds. And so the cells are differentiated. So for example, we have epithelial cells that are inside of our cheek and our mouth that are flat. They have a particular shape and function that's different from, say, muscle cells that are in our heart. So we have different kinds of cells, and because those cells have job specialization, they actually have to coordinate with one another and work together in order for the entire organism to be able to do all of its needed function. So those are our two characteristics of multi-celled life. So when we start to look at multi-celled life, we're going to be looking at eukaryotic organisms. Eukaryotic organisms are the only kinds that can be multi-celled. Prokaryotic organisms, which we're going to come back to in the next lab, like bacteria and archaea, can only be single cells or colonies. If you look at these different kinds of eukaryotic organisms, most of them you probably know already. And when we think about eukaryotic organisms, they're easy to classify into the major groupings of animals and plants and fungi. So take a minute and look at these pictures and decide which ones you think are animals, plants, or fungi, and if there's any other kinds of eukaryotic organisms that are in these pictures. Right, so you probably went for animal right away. Animal's easy. That's a hummingbird. It's a bird. It's an animal. Our earthworm is also an animal. And if you are interested in marine biology, you might already know that this guy is an animal as well. It's a sponge, and it has a really unique and simple body structure that we'll come back to later in the semester. It might look like plant-like to you, but it really is an animal. So when we look at plants, we have a lovely example of a rose plant here. And this organism down here that is underwater, it's actually not a plant. And the reason for that is its body structures are very different. And we're going to define the plant kingdom as plants that live on land. So this plant-like organism that lives underwater, it's an algae or a kelp, and we're going to put it into a different classification grouping. So it's not an animal, it's not a plant, it's not a fungus either. Here's our mushroom, it's a fungus. And the same thing with these other organisms down on the bottom of the slide. These are also eukaryotic organisms, but they're single-celled. So this one here is Euglena. It's a single-celled eukaryotic organism. So we can have single-celled eukaryotic organisms that don't belong in either the animal, plant, or fungi category. Same thing with this one here, that's Paramecium. It's a single-celled eukaryotic organism that's not an animal, a plant, or a fungus. And our kelp over here, this guy, is a multi-celled eukaryotic organism that's not a plant, an animal, or a fungus. So we'll come back to those in our eukaryotes versus prokaryotes lab next week. When we look at all different kinds of living things, we're going to be able to see that some of them come in a single-celled body and others are in multi-celled. If we look in a pond, like this example of a variety of organisms we might find from a pond, we'll see that there's a wide variety of sizes and that we can have single-celled, multi-celled, and colonial organisms here. I'll come back to the definition of the word colony in just a minute. But because we have this nice aquatic environment that's quite comfortable to live in, we'll see that the size of the organism doesn't necessarily tell us whether it's single-celled or multi-celled, because we have an amoeba here. It's a single-celled eukaryotic organism. And then we have rotifers here that are about the same size, but rotifers are multi-celled. They're complicated organisms with a complete digestive tract, tiny brain, and they're about 1,000 cells. So we have a variety of these tiny but multi-celled organisms that live in very moist or aquatic or marine habitats. And this size of the body overlaps with the size of single-celled organisms. So we can't necessarily tell just by looking at the size whether it's single or multi-celled at these small sizes. So the very, very biggest of cells is only up to about a couple centimeters. So if it's bigger than a couple centimeters, it's a multi-celled thing. All right, when we look at our plants versus our animals, you should already know the basic structures of plant cells and animal cells. Plant cells are typically going to look rectangular, like bricks. They have a cell wall, and that's why they have this more rigid shape. Animal cells have this irregular outline typically. They may be a variety of different shapes, but they don't have a rigid form like the plant cells do because they don't have a cell wall. Animals are going to be defined by being heterotrophs. That means that in order to get their food, they have to ingest and absorb it. So they're going to eat in order to get energy. Whereas plants are going to be able to do photosynthesis, and so they're defined as autotrophs or self-feeding. They don't have to go out and find something to eat in order to get energy. They just sit there and absorb sunlight energy and can turn it into potential energy stored in molecules inside their bodies. So we have these two characteristics already that are going to allow us to distinguish between plants and animals when we're looking at their cells. The difference between their shape because of cell wall versus no cell wall. And if they're live and fresh cells, we'll be able to see that the plant cells have this nice bright green color typically, which reflects their ability to do photosynthesis. Now these particular animal cells that are on the right-hand side here are epithelial cells from the inside of a human cheek, and we will come back to that activity next week. If we were in lab, you'd get to see your very own cheek cells, but you'll have to wait for another day, or maybe you did that already in Bio 120. Right, so the plant cells that we have on the left-hand side, those are from a plant called Elodia. And if you've ever had a freshwater aquarium, you might have this plant. It's also often called goldfish grass. And we're going to be able to look at these examples of a plant cells. It's not an algae, it's a true land plant that has gone back into the water. And if you have a really happy aquarium, it'll grow a little stalk up with little flowers on it. So you can tell that it truly is a flowering plant. We look at the way that organisms are constructed. Biology is broken down into the level of size of things that we're looking at. So if we start at the very smallest level of organization of the body of a living thing, it's made of atoms that are formed into molecules. And then those molecules are going to be organized into organelles inside the cells. And those cells are going to be organized into tissues. Tissues are organized into organs, and organs are organized into organ systems. And then we put the whole organism together by putting together all the organ systems and wrapping a wrapping around the outside of it, which is actually an organ system itself. So this idea that we have a hierarchy of structure is much like the idea when we were doing classification. We have a hierarchy of categories that our classification is going into. But for life, we have at each of these levels, when we put together a variety of tissues into an organ, the organ can do something that the tissues couldn't do by themselves. So when we have muscle tissue together with connective tissue and nervous tissue, and we form it into a structure that is the heart, that body organ has the ability to contract on a regular rhythm that those tissues couldn't do by themselves. So every time we go up a level, we see what are called emergent properties. And these emergent properties are things that emerge or become possible when we put together those simpler sub structures. So I like to think of it as when you have a team in sports, each individual could maybe run around the soccer field, you can dribble the ball to yourself, it gets kind of boring. And so you have an individual player. When we put several players together into a team, then we can have cooperation between different teammates. You can have strategies. You can kick the ball to one another. And so there's things that a team can do that an individual can't do. Now, even if you have your own team, that's still not much fun because you want to play against somebody else. So we're going to organize our teams into a league. And when we have multiple teams and we have an organized way in order to plan tournaments between them, then we can have a competition and figure out who's the best team for this season by having the emergent property of that organization of the league by taking multiple teams and putting them together. So that's the basic idea of how the bodies of multi-celled living things are organized. So we can't see individual cells for the most part with our naked eye. And we can sometimes see, though, a colony of individual cells with our naked eye. And a colony is different from a multi-celled organism because a colony is made up of individuals and they're living closely together. Often they're actually stuck together. But if you separate them apart, they can live separately from one another. They aren't differentiated and they aren't independent, or they're not interdependent. So if we pull those individuals apart from one another, then they'll just keep dividing and grow more. They aren't going to die. So if you imagine you sort of pulled your arm off, if it was done in the right way, then you would die. But if you take apart a colony of bacteria or fungi that are shown in the agar plate on the lower right-hand side, if we just take little swab of that bacteria and we smear it onto another plate, the original colony and the new colony are going to keep growing. So we have a couple examples on this plate. We have colonies of fungus. These are molds. They started from one spore and then grew by cell division to form a colony that's visible to the naked eye. So the cells aren't visible to the naked eye, but the colony is. Same thing with those bacterial colonies. There's about 1 million bacterial cells in each of those colonies. So one bacterial cell fell onto that dish and it started to grow and divide. And within a week, we see a little dot there that's a few millimeters across. It is visible to our naked eye. Each of the bacterial cells that are in that colony, however, is not visible to our naked eye. And there needs to be a bunch of them for us to be able to see it. Some other important kinds of colonies are one in the top right-hand corner. Choanoflagellates are actually a group of flagellated single-celled eukaryotes that tend to form together into colonies. And that sponge animal that we looked at a few slides ago, its body structure is very similar to this. So we think that the choanoflagellates are actually the ancestor of all animals. In the lower left-hand slide, the cyanobacteria, they often form these long chains. And so even though they're bacteria, they're prokaryotic cells, we can often see them in large mats in ponds, and they're visible to the naked eye because they're brightly colored, their cells are fairly large, and they form these colonies. Sometimes we talk about a colony of honeybees or a colony of sea anemones, and those are also individuals that are living together. But if you take one honeybee out, it doesn't immediately die. Now you would have to take care of it, but we sometimes use that word to describe a group of animals living closely together as well. So when we look at how we define the groupings within the eukaryotes, there's a traditional way that we did this, but DNA sequence data has shown more recently that this was inappropriate, that this group we used to call protists is actually polyphyletic. It's not monophyletic, that we have things that we used to call protists that actually are more closely related to animals, and different ones are more closely related to plants, and different ones are more closely related to fungi. There's been a big reorganization of how we think about single-celled eukaryotes in the last 10 years or so. Our definitions of fungi, plants, and animals still apply. We're going to use 3 categories that are going to allow us to distinguish between our fungi, our plants, and our animals. Fungi can be single or multi-celled. Single-celled fungi are things like yeasts. Multi-celled fungi are things like mushrooms. Plants, we're going to define as being multi-celled. Animals, we're going to define as being multi-celled. And all the leftover stuff, what we used to call protists, is going to include some single-celled and some multi-celled organisms, not typically in the same taxonomic group. We're going to look at these organisms and see if they have a cell wall or not, and what the molecule that the cell wall is made of. So fungi are going to have a cell wall made of chitin, plants are going to have a cell wall made of cellulose, and animals have no cell wall. Protists can have a variety of different types of cell walls or none, depending on the taxonomic group, and a variety of different molecules. So we'll come back to those later. We're going to define each of these groups as well by their energy mode. That means how do they get their food? We already talked about animals being heterotrophs and plants being autotrophs. Fungi are also heterotrophs. So if you think about a mushroom, it's kind of whitish or gray. It's not bright green like a plant is. And so it's actually also absorbing its food from the environment. It doesn't have a mouth, it isn't going to eat, but it is going to absorb molecules across the cell wall. Protists are an incredibly diverse group. We're going to have autotrophs and heterotrophs in different taxonomic groups within protists. Now, when we look at their life cycle, you're going to talk about this in lecture. So when we have sexual reproduction in these groups, you're going to see that there may be a multi-celled individual that is haploid, that is diploid, or that they might have both in the life cycle. So we'll come back to those categories later. So the way that taxonomy has traditionally divided up these eukaryotic groups, we now have a change. So we definitely have prokaryotes, and off here on this cladogram that's showing bacteria and archaea split off from this tree and evolved on their own. They stayed prokaryotes. Somewhere on this tree, we have the evolution of the eukaryotic kind of cell. And all of the organisms that have a eukaryotic kind of cell have a common ancestor. So we've split the eukaryotes into 4 major taxonomic groups that are quite different from one another. So these are actually in between domain and kingdom. So different textbooks, different authors call them different things. I'm going to call them supergroups because that is one way that we can decide what we're going to call them. And we have different levels within these before we get down to kingdom. So it gets a little complicated, but we'll try and use the same vocabulary all the way through. So eukaryotes are now divided into these 4 main groups: the excavata or excavates. The clade called SAR is made up of 3 groups called the straminopiles, the alveolates, and the rhizaria. So it's sort of an acronym for those 3 groups. Then we have the uniconts, and the uniconts includes both fungi and animals. You can see that this category is a bigger category than kingdom. And the archaeplastids includes the land plants as well as green algae. So the land plants, we're going to start referring to them as the embryophytes, and that's going to equal plants. We're going to quickly look at the basic body structure of these different multi-celled groups: the fungi, then the plants, then the animals. Fungi have no body organs. They are incredibly simple, yet incredibly effective at living their lives. And they probably were the first organisms that came into the terrestrial environment. Fungi have single cells that make up their bodies, and these cells are called hyphae. If there's only one of them, we'll call it a hypha, not a hyphen, like the little dash you put in between different parts of a word. So here's a hyphen, right? But those cells are called hyphae, just a hypha if there's only one, or hyphae if there's a group. It's hard to single one out, so usually we'll say hyphae. Now, when we have an individual fungal organism, we're going to say that its body is going to be called the mycelium. So when we say that, that's the word we're going to use for the body. So it's going to have an extensive underground network of these fibrous cells that are in a mesh-like structure, like a scrubby sponge that you might have that's made out of a mesh of plastic at your kitchen sink. And those are going to be intertwined and go throughout the soil. So remember that fungi are heterotrophic. And so they're going to actually be secreting digestive enzymes outside all of these hyphae. That's going to dissolve and disintegrate any organic matter that's in the soil around those hyphae, and then they're going to absorb those digested molecules straight from the soil. The structure at the top that is a spore-producing structure. This thing, we're going to call it the mushroom, and it is actually a reproductive structure. So like you see a fruit on a plant, and that fruit is for reproduction, the mushroom that's growing up out of the ground is not the whole individual. There's an extensive individual that's underground, but that mushroom is just sticking up to the surface, and it's going to produce the reproductive structures called spores. So if you think about it, the reason why it's nice to grow that structure up from the ground is that it's going to do a better job of dispersing those spores so that it can colonize new environments. When we look at other kinds of fungi, we don't just have mushrooms. We have yeasts, molds, and mildews. So we'll come back to that. We're going to have a whole lab just on fungi. When we look at the plant body, it's quite different. It's more complicated than fungi, not as complicated as animals. And we're going to focus today just on the very basic terminology for defining the different parts of the plant body. Plant body organs come in three kinds. So when we think about animal body organs, we have many, many different kinds. We have the heart, we have the lungs, we have the brain, we have the eyes, we have all kinds of different things. Plants only have three basic body organs: roots, shoots, and leaves, right? So those are our plant body organs. The structure of the leaf is flat. It has a wide surface area and it's being held up by the stalk. So that's its shape. And often they can be quite large and they tend to be angled towards the sunlight. And the function of the leaf is in order to absorb sunlight and do photosynthesis. The function of the shoot, it holds up the leaves. And it's actually going to be really important to transport substances from the leaves to the roots and from the roots to the leaves. So it has a structural function and it has a physiological function as well. And the roots are going to also have a structural function and a physiological function. Their structural function is to anchor the plant. Those roots interweave themselves into the soil and make sure the plant doesn't fall over. And they're also going to absorb water and nutrients from the soil. So when we look at the basic body of a plant, the things that it has to do, it's going to be absorbing things that it needs into the roots. Those are going to get transported up the stem. The leaves are going to be taking in CO2. They're going to be putting out oxygen. They're going to be producing sugars. I'm going to draw glucose here. And the glucoses, the sugars are going to get transported to the rest of the body. The roots have to get fed by the leaves. So that transport is going to go both up and down. All right, so when we look at all of the different kinds of plants, they just have these three simple body types, but there's an amazing diversity of body forms of plants. So we're going to look at the four basic kinds of plants. If you look at this picture, it's just overwhelming, but we have every range of things here. We have grass down here. Looks like we have some ferns. We have some, maybe we have a shrub here. We have some little flowering plants down here. We'll learn the name that we're going to use for those. And then we have these larger plants up here. We can see their stem, and we're going to call those, trees. And so that's what I'm talking about. We're going to try and make a categorization of different plant body types or body forms. And we're going to have plants that either are only green growth, they tend to only live a year. And we're going to have plants that live for longer than a year, and they're going to have some kind of woody or brownish and hard structures. Their stems are going to get much thicker, and they're going to have a body that's made with, reinforced with wood, right? Wood is that brown, hard stuff that we can burn in a fireplace. So these hardened bark covered stems are called secondary growth because they happen in the second year or more in the plant. We have a tree, we're going to define it as a large single stemmed plant. And typically it isn't going to have any leaves near the bottom. So if we look at this region, we can see that it has this canopy up here where all the leaves are. And we can see that it's a tree because it has a distinction or a separation between where the leaves are and then this trunk at the bottom. A shrub or a bush is going to be a medium sized plant. It's going to have more than one stem. So we have these different branches that are growing. It's going to have leaves that go all the way to the ground. And if we peek into it, if we can see the actual stem in the middle, we're going to see that it's brown, and that we may have small shrubs or larger shrubs. We may have shrubby trees and tree-like shrubs, but we're going to try and keep it simple for this class, and we want to have some obvious differences here. We look at plants that only have primary growth. That is, their bodies are basically all green. They're kind of soft and fluffy. You can literally imagine biting into them. That we're going to have plants that sort of grow up with obvious leaves on them, and we're going to call those forbs. Herbaceous plants is the opposite of woody plants. Their bodies are made only of this sort of green, soft, fluffy growth. They don't have any wood or that secondary growth. So all the plants that we use as herbs, like basil and chives and parsley, cilantro, these are all herbaceous plants. Now the ones that have long, thin leaves that grow in a clump are going to be grasses. Grasses are actually a taxonomic distinction. And that when we look at this particular group of grasses, they have certain growth characteristics. So they're going to have long, thin leaves. They're going to grow from a base where all of those leaves are coming up from the ground together. All right, so I'd like you to be able to recognize the difference between these different growth forms, and we'll have some practice in lab today. When we look at a plant that's not a grass, we'll see that it has different ways of organizing the leaves onto the stem. So we're going to have a basic stem that is growing up the shoot. And then if we have a little stalk, which is actually called petiole, and then a single leaf is sticking out from it, we're going to call that simple. And the other thing that can happen is that instead of having this little petiole sticking out and it only has one single big leaf on it, it can have more than one leaf sticking out from it. So in that sense, we need to make a distinction. So we're actually going to call this a leaf. And when we have this compound leaf, we're going to call that a leaflet, little subleaf. But this whole thing, the thing that sticks off from the stem of the plant, the whole thing, we're going to call it a leaf. All right, so it's a compound leaf because the whole leaf is made up of multiple leaflets. All right, botany is fun. When we look at the stem of the plant as well, the main stem of the plant, we're going to look and see how those leaves are growing off of it. So this example over here is a plant with simple leaves. And in this case, they're growing off of the stem at the same height. So we say they're opposite. The two leaves are opposite to one another at the stem. In this plant, the stem connections to the leaves are stair-stepped and they happen to be on opposite sides from one another. So we call that alternate. And this is a very important way to tell the difference between different groups of plants. So we're also going to look at that characteristic and practice that. When we look at all the different kinds of plants, another major characteristic that's going to be distinguished between them is the monocot plants versus the dicot plants. So the flowering plants, the enormous group of plants with a huge diversity, the two major groupings within that you can easily learn to recognize by eye when we look at a plant are the monocots and the dicots. They get this name because the embryonic leaf that's inside of the seed is called a cotyledon. And when the seed germinates and the plant starts to grow up from the ground, monocotyledonous plants have one baby plant leaf or cotyledon inside that seed. And so when they germinate and start to grow, you're going to only see a single leaf sticking up. I've got a big palm tree in my yard, and when those little palm trees start to grow, there's a single leaf sticking up. So I have to pull them out. I don't want a palm tree, you know, right next to my trash cans. I don't want a palm tree in the middle of my vegetable garden. So I see a lot of those. A dicot has two cotyledons, and so when it grows up from its seed, we have a pair of little leaves sticking out opposite to one another. Turns out they have also other structural characteristics that we can see quite easily, even if the plant doesn't have any flowers on it. We'll come back to looking at the seeds in a few labs. When we look at the leaves of a monocot, we'll see that the veins or the vascular tissue is going to be parallel, and they tend to have long, thin leaves that we can see those parallel veins on. In dicots, the veins are going to intersect one another. So we'll see this branching pattern, right? The opposite of parallel. And they also tend to be broader. They don't tend to be long and thin. And there's a whole variety of different shapes. So if we have a plant with compound leaves, then each of the leaflets will have this pattern. Now, if it does have a flower on it, we can see that the petals of the flower are going to be in multiples of 3 for monocots. That makes it really easy to tell if we can see the flower. If it has 6 petals like this lily flower, we know it's a monocot. And then the dicots, we're going to see that the flower either has 4 or 5 petals. Now we have all kinds of crazy flowers that it's not easy to tell on, but the very basic simple patterns. So monocots are going to be plants like lilies, tulips, grasses such as corn or bamboo, palms, orchids. And there's nowhere near as many monocot plants as there are dicots. So dicots are everything else. It's going to be beans, oak trees, fig trees, basil plants, sunflowers, daisies, just about anything else that you can think of. So if you have absolutely no idea what the plant is, if you guess dicot, you have a good probability that you're right. All right, we're going to look a little bit at the structure of plant stem in this lab. So we're going to have a plant stem, and we know it needs to do transport. So it's going to have a way to get a liquid with substances dissolved into it going up and down through the plant. So it turns out that it actually has a system of pipes that go through the stem. And we're going to call these pipes vascular tissue. And we're going to think about right now what that's going to look like when we cut a cross section across the stem. So this is going to be a cross section. And if our stem is round, it's going to look round. And we're going to be able to see that vascular tissue in some way in the stem. Now, if we take a longitudinal section, or we cut down through the stem a long ways, right, we're going to get more of a rectangular shape. And we're going to see this pipe, if we happen to have sliced through it, we're going to see that it looks just like a long thin rectangle. So when we look at different slices or sections through the body of our plant, we're going to be able to see the structures. Most of the time, we're going to look at a cross section. So today we're going to do an activity. We're going to be able to take a cross section of a plant stem and look at what kinds of tissues that it has inside of it. An herbaceous eudicot stem is just a way of saying it's one of those forbs or sort of fluffy plants. We aren't going to see any secondary growth or woody stem here. And plants, again, are simpler than animals. They have 3 different kinds of tissue. They have dermal tissue, they have ground tissue, and they have vascular tissue. The vascular tissue we're going to see occurs in these groupings or bundles. And the function of the vascular tissue is transport. And the structure of it is that it's these long, open, thin tubes. That works really well for transport because we have these long, thin tubes. It's actually going to allow the flow of that body fluid. So there's a body fluid inside of the tubes. It's called sap, right? So it's like blood. This body fluid of the plant is going to be able to move from the roots to the leaves or the leaves from the root because these vascular tissue cells are long and thin, and so they connect different parts of the body together. The dermal tissue is always going to be just this one layer of cells in our eubaceous or our herbaceous eudicots. And it's going to be very simple cells, and they're going to be forming the surface, and their function is protection, like our skin. Everything else in plants is ground tissue. So if it's not vascular tissue, it's not dermal tissue, it's ground tissue. And there's a variety of different types. It can be structural, it can be for storage, and so we're just going to say everything else for its functions. And we'll come back to that more when we go into plant labs. But basically, every other place where we see any cells that are clearly not vascular tissue or dermal tissue, those are going to be ground tissue. Now moving on to animals, we're going to look at same thing. How do we cut through their bodies, and what do we see when we take these different sections? So it turns out that animals can have different kinds of body plans, and we're going to define those as how we can slice the body of the animal in half in order to get two sides that look the same. So animals can either have this radial body plan or a bilateral body plan, and the way we're going to describe that is as symmetry. The only kind of animal that can be asymmetrical or irregular in shape is that sponge animal, and we'll look at some examples of it later. We have, when we look at all the different kinds of animals, we only have two kinds that have radial symmetry. We have cnidaria, and we have echinoderms. So echinoderms are things like a starfish or a sea urchin. So either their body has a circular shape, so our cnidaria, this is an example of a cnidarian, and its body is basically a column. And it has these tentacles sticking out from it. Its mouth is going to be in the middle here. And if we slice through it anyway, we'll get the same thing on both sides. So starfish, we have to slice in a little bit more particular way, but there's more than one way we can slice through it, and we get the same thing on both sides. Right, so that's going to be our defining characteristic of radial symmetry. When an organism is radially symmetrical, it can go any direction in the environment and be equally aware of or able to respond to it. When we look at a bilaterally symmetrical animal, it's going to distinctively have a head end with sense organs, and it's going to definitely be having one particular direction that it's going to prefer to go. So if you see an animal that has a head in any way, shape, or form, it is bilaterally symmetrical. When we look at the body of our radially symmetrical organism, just like the plant, if we're going to take a cross section, it's going to go across this way. If we take a longitudinal section, it's going to go that way. Because the animal body is a little more complicated, there's actually 3 ways we can cut through it. If we're going to take a cross section, we can actually take a variety of cross sections through the body, and they're going to look different from one another. So these cross sections, sometimes we call it a transverse plane through the body, but if we take a slice of the animal that direction, we'll see what's inside of it. And if we stack all those together, then we can actually see basically the inside of the whole body. Depending on the animal, we might also want to take a slice through it from the top, going the long ways. And that's going to slice in our fish right through the spine. So we'll sort of see the inside from the middle if we make that slice. And we call that a median or sagittal plane, but it's a longitudinal section. Now we don't use this other section very often, the frontal plane. That would be like slicing sort of down through the top of our head, through both of our ears, and then through both of our shoulders. It would basically separate our body from front to back. The only application we have of that often in biology is when we're actually imaging the brain, because we're oriented in an upright posture. Frontal sections are the way that we slice through the brain, basically at a different angle than we would through other animals' bodies. The other thing we're going to look at for animals is what kind of appendages, what kind of body parts do they have dangling off of their main body. So this animal up here has tentacles. The fish here is going to have fins. Humans have arms and legs. We might call all four of the structures dangling off your dog, we might call them legs. And so we might look also at a worm, and the worm body is not going to have any appendages at all. So we're going to have some practice doing that today too. In animals, the body is much more complicated than in a plant. You may not be incredibly interested in plants, but once you start studying them, you will be very excited at how easy it is to study about plants because they're so much simpler and they're nice, and there is an elegance to the way that their bodies are able to function without all of the crazy kind of complexity that we have. So when we look at animal bodies, we're going to put the tissues that they have into 4 categories. So plants have 3 tissue types, animals have 4 tissue types. Epithelial tissue is going to be on the surface of the body, whether that surface is an internal surface like the inside of the chamber of your heart, or whether it's an outside surface like the outside surface of the heart. If it's a surface, it's got epithelial cells. So they're directly comparable to the dermal tissue cells in plants. And epithelial cells have a structure that is often stacked or layered, or they're very tightly packed together. So we'll see a few examples of epithelial cells. They're fairly easy to observe because they have large nuclei and there's a nice pattern to them, and where they are is really easy to find them. You just find any surface and the epithelial cells are the ones right on the top. So their structure is some kind of packed together cells in a little row or layers of cells on top of one another, and their function is protection and separation from the rest of the body, just like plant. The next kind of tissue is muscle tissue. It's made up of fibers that contract. So there are proteins inside those muscle cells that are able to slide relative to one another, and that shortens the muscle cell. So they have this striped appearance. They're very densely packed, and their function is to contract or get shorter. So once they're fastened to some body part, if they contract or get shorter, then they move that body part. So we think of their function as being maybe to do locomotion or to do motion in general, but their function is just to contract, and then whatever fastened to them ends up moving. Muscle tissue is also relatively easy to learn to recognize in histology slides, right? We also have some places we expect to see it, so that also helps us be able to find it. The third kind of tissue is nervous tissue. Nervous tissue doesn't look like much when we have a slide preparation of it. We will sometimes know that there's a nerve cord going through the body, and so we know that that's made of nervous tissue, but nerve cells are hard to see in histological preparations. They have long extensions on these nerve cells that are going to connect to one another. And so their shape is that they have these long, thin projections that are connected to other nerve cells in such a way that they can pass electrical signals through the body. Now, that is the way the body does communication of information from one place to the other. So although their overall function, their bigger picture function, is to cause communication to happen between different body parts, each individual nerve cell, all it knows it's doing is it's transmitting an electrochemical signal from one end to the other. The last of the 4 tissue types is connective. And again, just like ground tissue in plants, everything else is connective tissue. So if you want a definition of connective tissue, it is that it's a sparse or a thin amount of cells with some space and stuff between them that is not made of cells, and that allows us to apply that description to every other kind of tissue that isn't epithelial, muscle, or nervous tissue. So we see that there is some kind of material that those cells secrete around them. For example, in bone, there are cells that make up the structure of the bone. The minerals that are deposited that make it, you know, that white chalky hard stuff that's left after we die, those that material is secreted by those bone cells around them, which is why that if you have a broken bone, it can heal because the cells get busy and they start secreting more material in order to repair it. Connective tissue is also in things that we think of like cartilage and ligaments and tendons, and those have kind of a gelatinous matrix that has cells embedded in it. So there's not that many cells, and that's why it takes a lot longer for a damage to cartilage to heal than damage to muscle, because those few little cells have to work really hard to try and repair that structure. The weirdest one to me is blood. Blood is a tissue. Even though it's a liquid, it has a sparse grouping of cells in it, the blood cells, the white blood cells and red blood cells, and then the stuff, the material that they are in is a liquid, plasma. So they don't necessarily secrete all of the material that's in the plasma around them. Your kidney does part of that job, and your liver, but blood itself is a tissue. So not everything that you think of like a Kleenex tissue is actually a solid three-dimensional structure. A tissue can be a liquid or a gel. All right. So when we're looking at slides today, I want you to focus on the epithelial tissue and the muscle tissue, because those things are really easy to see and obvious. Certain kinds of connective tissue are also easy to notice. So we're not going to worry too much about nerve tissue. One of the slides that we're going to look at is of an earthworm today. So earthworm is a sort of a long, squiggly worm-like animal. We're going to take a cross section through the body. It doesn't have any arms or legs. And then we're going to see a slide that looks like this. So this is the inside of the gut. Here is where food is going to go. And then we're going to see that we have a nerve cord. So if you want to look at nerve cells, you're going to be seeing them. They're going to look like a bunch of bundles because you're looking at a cross section of that nerve cord, and those axons that make it up are long, thin tubes. The animal has a body space inside of its body. And so this area called the coelom, it's empty space because there's empty space. But I want you to focus on any part of the body wall. And you're going to be able to see on the very outside, there's epithelial cells. On the very inside, there's epithelial cells. There's going to be this region that's all muscle tissue, and there's going to be some connective tissue in between them. So if we look at the inside of the intestinal cavity back here, there's an epithelium here, there's an epithelium there. There's muscle tissue around the gut as well. And so you're going to be able to see muscle tissue that's going in two different directions. If you notice up here, it says circular muscle versus longitudinal muscle. Circular muscles actually go like a rubber band around the body of the worm, and when they contract, the worm gets skinnier. So we're looking at those muscle fibers from the side. So we're looking at the fibers are actually going this direction. And when we look at longitudinal muscle, that's going down the length of the worm. And so what we're seeing, if we see this cross section here, we're seeing a bunch of these muscle cells, muscle fibers packed together. So it's going to look more like a bundle of straws. All right, so I'm not going to expect you to be able to identify every single kind of tissue we see on this slide, but it's just to start to give you an idea of when you look for the epithelial tissue and the muscle tissue, you can easily get oriented to these different structures. Histology is quite challenging. It takes a lot of practice and a lot of different types of examples to look at, and we don't have time to do that, so we're going to keep it simple. Now, a cool thing about earthworm is that it has these little bristles that it can extend. It can poke them out, these setae. It's got a pair on the very side, and it's got a pair kind of on the lower side. So as soon as you can see those in the slide, it has little muscles that it can use to pull them back inside and to poke them out again. Most sections, slide sections that we see of animal tissue because it's a whole animal that we're looking at, it's actually going to have muscle tissue, it's going to have connective tissue, it's going to have nervous, and it's going to have epithelial tissue. So you're going to have to, you'll see all those different kinds. All right, so if you're ready to go look at the slides for this activity, there's a few slides and a few videos for the activities we're doing for this lab. So I hope you are excited about making sure you know all the vocabulary and clearly can define the body types, tissues, cells, and organs of plants, animals, and fungi.